400 million years ago, something grew to 8 meters tall on a planet where nothing else on land stood higher than your knee. It wasn't a tree. Trees wouldn't exist for another 30 million years. It wasn't a plant at all. For over a century, the best guess was that it was the largest fungus to ever live. But when researchers finally ran chemical tests on the fossils in 2026, the results didn't match any fungus, living or extinct. They didn't match any plant or algae either. Whatever Prototaxites was, it appears to be something science has never seen before or since. To picture the world where Prototaxites lived, you have to forget everything you know about land. No grass, no flowers, no leaves rustling in the wind, because leaves hadn't been invented yet. The Silurian and early Devonian landscape was flat, wet, and covered in thin crusts of moss, liverworts, and bacterial mats. The tallest vascular plants on Earth were stubby little stems, most of them barely reaching your ankle. A plant called Cooksonia, one of the most successful species at the time, topped out at roughly 6 centimeters. Picture the smallest shrubs you've ever seen, then cut them in half. That was the competition. The animals were equally tiny. Mites picked through the moss. Scorpions smaller than your thumb hunted between the liverworts. Millipedes were among the larger creatures crawling across the ground. Nothing with a backbone had left the water yet. Vertebrates were still confined to rivers and shallow seas. It was a world without birdsong, without footsteps, without anything tall enough to cast a shadow. And then, rising out of this carpet of green, Prototaxites stood like a pillar, 8 meters tall, roughly a meter wide at the base, tapering slightly as it reached upward. For tens of millions of years, these organisms were the tallest things on dry land, not trees, not even close to trees. Trees were still deep in the evolutionary future. These columns stood alone on an otherwise flat horizon, surrounded by moss and millipedes and not much else. The first person to study Prototaxites fossils was a Canadian geologist named John William Dawson. In 1859, Dawson examined specimens that had been collected from the Gas Peninsula in Quebec, and he was convinced he was looking at the remains of a giant conifer. Partially rotted wood, he wrote, with a loose tissue structure and tangled internal fibers that resembled fungal decay. He named the organism Prototaxites, meaning first yew, because he believed it belonged to the same family as yew trees. To Dawson, this was simply a very old, very large tree trunk that had been colonized by fungi after it died. 13 years later, a rival scientist named William Carruthers looked at the same fossils and came to a completely different conclusion. Carruthers argued that Dawson had it backwards. This wasn't wood at all, he said. It was an enormous alga. He rejected Dawson's name entirely and insisted on calling it Nematophycus, meaning stringy alga. The two men disagreed publicly and bitterly, and the debate over what Prototaxites actually was had officially begun. That disagreement would last for more than 140 years. Over the decades, different researchers classified Prototaxites as a land plant, a brown alga, a red alga, a green alga, a lichen, and 3 different types of fungus. Nobody could agree because the internal structure of the fossils didn't quite match any of those categories. The tubes inside Prototaxites were arranged in a complex layered pattern looked, but also vaguely plant-like. And depending on which researcher you asked and what angle they cut their thin sections. Then in 2007, a geochemist named Kevin Boyce at the University of Chicago tried something nobody had done before. According to a study Boyce published in the journal Geology, his team measured the carbon isotope ratios inside Prototaxites fossils and compared them to the ratios found in plants from the same time period. If Prototaxites had been photosynthetic, like a plant or an alga, it would have pulled carbon from the atmosphere in a predictable pattern. Instead, the carbon values were all over the place. They varied wildly from specimen to specimen, which suggested that the organism was feeding on whatever organic matter happened to be nearby, not making its own food from sunlight. That finding pointed strongly toward a fungus, a heterotroph, something that consumed other organisms rather than photosynthesizing. Boyce's team concluded that Prototaxites was a giant fungus, and for nearly 2 decades, that became the accepted answer. The mystery, it seemed, had been solved. But Prototaxites wasn't done confusing people. In January 2026, a team led by Corentin Lauren published a study in Science Advances that upended the fungus classification entirely. Lauren's team examined fossils of a species called Prototaxites taiti, preserved in the 407-million-year-old Rhynie Chert deposit in Aberdeenshire, Scotland. The Rhynie Chert is one of the most important fossil sites on Earth for understanding early land life, because organisms trapped in it are preserved in extraordinary detail, right down to their cellular structures and chemical signatures. Lauren's team used infrared spectroscopy to analyze the molecular fingerprint of Prototaxites and then compared it to the chemical signatures of fungi, plants, arthropods, and bacteria preserved in the same rock. According to their paper in Science Advances, the results were clear. Fungal fossils from the Rhynie chart all preserved chemical markers associated with chitin, the tough polymer that makes up fungal cell walls. Every known fungal lineage, from mushrooms to molds to the most ancient basal groups, produces chitin during at least part of its life cycle. Prototaxites had none. Zero chitin signature. The molecular fingerprint didn't match any fungus the team tested, living or extinct. And the structure was equally strange. The interior of Prototaxites tidy contained 3 distinct types of tubes, including large, thick-walled tubes with a kind of banding pattern and dense spherical regions called medullary spots. None of these features matched any known fungal anatomy. If you're enjoying this deep dive into Earth's strangest organisms, hit subscribe. There's plenty more where this came from. The team also ran their chemical data through machine learning classifiers trained to sort biological specimens by kingdom. The algorithm couldn't place Prototaxites with fungi. It couldn't place it with plants. It couldn't place it with algae or bacteria either. The organism fell outside every category the classifier had been trained on. Loren and his colleagues concluded that the morphology, metabolism, ecology, and molecular fingerprint of Prototaxites preclude its placement in any known crown lineage of multicellular eukaryotes. In plain language, this thing doesn't fit on the tree of life as we currently draw it. Their best assessment was that Prototaxites belongs to a previously undescribed, entirely extinct group of complex life forms, a branch of the evolutionary tree that produced nothing else we've ever found. Think about what that means. For roughly 50 million years, the tallest organisms on land belonged to a lineage that left no descendants, no cousins, no surviving relatives of any kind. They dominated the landscape and then vanished completely, leaving behind nothing but fossils that would confuse scientists for the next 160 years. Part of what makes this so difficult is that we have different reference points for what life looked like during the Silurian and Devonian. The world was genuinely alien. The atmosphere had more carbon dioxide and less oxygen than today. Land was colonized by a thin film of simple organisms. Into that world, Prototaxites grew to the height of a 2-story building, and we still don't know how it fed, how it reproduced, or what finally caused it to go extinct. One possibility is that the rise of true vascular plants pushed Prototaxites out. By the late Devonian, the first real forests were spreading across the continents. Trees in the genus Archaeopteris reached heights of 10 meters or more, and for the first time, something else on land could match Prototaxites in size and eventually surpass it. If Prototaxites depended on consuming the simple organisms in its environment, the transformation of that environment by larger, more competitive plants may have starved it out. But that's speculation. The fossil record doesn't give us a clear cause of death for the entire lineage. The other uncomfortable possibility is that Prototaxites represent something we don't have a category for. Not a plant, not a fungus, not an animal, not an alga. Something else entirely, a form of complex multicellular life that evolved its own solutions to the challenges of living on land and then disappeared without passing those solutions on to anything else. We tend to assume that the major branches of life on Earth are the ones we can still see: plants, animals, fungi, and a handful of microbial kingdoms. Prototaxites suggest that the story is messier than that. There may have been entire kingdoms of complex organisms that rose and fell before the tree of life settled into its current shape, leaving behind no living evidence that they ever existed. The only traces are locked in rock, waiting for someone to cut a thin section and hold it up to the light. Dawson looked at those fossils in 1859 and saw a rotting tree. Carruthers saw a giant seaweed. Boyce saw the world's largest mushroom. And Loren's team, after 160 years of debate, saw something that doesn't have a name yet, a branch of life that exists nowhere on Earth today. If you want more stories about the strangest organisms science has ever found, you'll find them on the channel.